Here we are up in the fifth floor of the west wing of the Natural History Building, and I'm going to talk to Melinda Peters, who is a museum specialist, and her job is the handling and curation of incoming and outgoing specimens. And are you going to show us how to mount a plant here?
Yeah, I'm going to show you kind of our technique. So things come in to us from all over the world, and normally it's just they come in their original newspaper. So they're pressed in the field. They've been frozen. We follow an integrated pest management where we freeze everything now. And then the volunteers and some staff, we work in this room. We have setup of mounting stations where we prep everything for the final product of a traditional specimen. So everything gets a label. Everything gets a fragment packet. Sometimes extra fruits are put in baggies. And our technique is a little different. We do a layout where the label goes here and the specimen is arranged in a way to show all characters, both sides of leaves, fruits or flowers that might be important. And then we only use a gummed linen tape and thread to secure the specimens. We don't actually glue the plant onto the sheet. We use glue for the label and the packet, but not for the actual specimen. And that technique's nice because it's a reversible process, and if researchers or curators like Paul need to access, something that's on the back of a leaf or something, if it's not arranged the way that they want it to, then we can redo it. So it's a really nice method that I think secures things better. And with DNA work, we are able to take leaf samples a little bit more easily.
Not my game is to ask the dumb questions.
Dumb questions? Sure, yeah.
So why the size?
Why the size of the paper?
Yes.
Well, this is a traditional size. So most collections all over the world use this 11.5 by 16.5 paper, and it also fits perfectly in the cases that we have.
Oh, yeah.
But this is a traditional. That's a good question.
And the content of the paper, is it set up
everything's archival. So all of the supplies that we use are made with archival quality linen and rag paper or the threads, and has like a neutral pH level. The glue that we use has a neutral pH, so we hope that long term it won't yellow or deteriorate as fast.
I'm going to give you a toughie here.
Oh, great. Okay,
good. Do you know possibly what may be the oldest herbarium specimen?
Here, well, we might see a few downstairs. It looks like we have something from the 1770s and maybe something between 1594 time period. But that's a good question. We may not know yet because we haven't totally digitized, or we don't, if it's in a case and been tucked away for a long time and hasn't been seen, then we may still find something out there. So let's go take a look.
Okay. Melinda's going to continue explaining about the Smithsonian and National Herbarium. And we're going to have two people be her students, so to speak. And we have Gabrielle David and Andrew Blackburn, who are working with us also. And but being that one's a fluvial geomorphologist and the other one's specialty is in soils, even though Andrew knows plants, I thought they should know more about plants, so Melinda's going to use them as her victims. So
Thank you very much. So now we're down in the kind of the middle of one of our floors here, and you can see behind us cases. So Bob asked earlier, why are they a certain size? Well, here you go. They fit in these cases, so that's one reason. How do we organize? We here at the U.S. National Herbarium have, we believe, over 5 million specimens. So that's a lot to keep, track of. We do that very well because we organize things by plant family. We use a kind of an old traditional system where families were grouped together in a systematic, a classification, taxonomy kind of way. Nowadays, we're trying to move towards a more current method of the angiosperm phylogeny group, but we'll see that's 5 million things that we need to move around. But we can find things because once you get to a family, then it's by the genus name, and we divide the world into regions because we are a worldwide collection. We have things from every nook and cranny, all over the place. Of course, we specialize in groups because curators that have come here from the past to now work on particular plant families or particular regions of the world. So, as with all collections, even your own that you might make, you're going to have either something local or something of a place of interest to you. And nowadays, the scope of these types of collections is really changing and is becoming more broader. So you all may have a use for this type of collection someday in your work. So let's just quick take a peek so you can see what a case looks like. All of the specimens are traditionally just stacked on top of each other in folders, and everything's labeled, so it's very easy to find stuff if you know what species you're looking for. Now, I don't know, so you may be looking at this and thinking, well, what about, large, bulky specimens or things that require maybe a different type of preparation? Well, we have that too. And so we can prep things in bulky collections, and we'll show you some examples of that in just a minute. Now, how did we get started? Well, when the U.S. was being explored in the 1800s, there were many collections going out. I have a list here of some of the collections that are represented in this group. So the United States Department of Agriculture, they were going out, they were sponsoring trips, so they were interested in the Mexican boundary survey. Lewis and Clark went out. Those specimens are at the Philadelphia Academy of Sciences. But we have things from the U.S. exploring expedition, the Mexican boundary survey, like I said. Here's an example of one of those. So these are some of the old traditional or old foundational specimens of our collection. And we continue to grow because of collaborations with other institutions like this all around the world that send us material from expeditions. And that's how each part of the collection grows. So let's take a look at a few other things. So here we are with, as I mentioned, some of the bulkier things that do not fit on sheets. We have a pretty amazing bulky bamboo collection because there was a curator here in the past interested in this group. So, how do you store something like this? It's a little bit easier than storing a whale skeleton, but we still have our challenges. So what else do we have? We have things like cacti here. So oversized cacti that can also be made into sheets, but sometimes you still need to preserve the whole organism. And then one of our favorites, of course, is the double coconut. So I don't know if somebody wants to see how heavy this is. This is the double coconut. It's really heavy.
Whoa.
It's endemic, which means it only grows in this one particular area, to the Seychelles Islands. Thank you. And you can imagine the names that it's been called. Here's it cut in half so you can see the inside. So it's the largest seed in the world.
Okay.
So now we're here with a type specimen, and types are the specimens that the plant name is based on. So when something new is collected in the field or when something is described, to get a species name, you have to have a specimen to base that on. The morphological characters, the genetics come into play, and that's how we come up with new species. So we're still finding things nowadays, and anywhere really. And there's still things in the U.S. that are new to science. So this is a really good example. This is just an image of the original specimen that's housed in another collection, but we have a fragment of this type, and this is a Lewis and Clark specimen. So you can see a tiny little fragment from the original collection. So we have a copy of that.
Why do you need a fragment of the original?
Well, because there's still characters there, and type collections are the most important collections because that's what the name is based on. So when researchers are studying groups, they need to be able to see a little piece even of the original source material. And something just that small would be enough to help somebody understand the morphological characters of that name, what was tied to that name. So, yeah, it's pretty neat.
So earlier you were uncertain if you had any specimens from the Lewis and Clark
yeah,
expedition.
that's true, yeah.
Can you explain why
Yes.
it's hard to find from a specific expedition?
So we were able to look up in the database. You see that this has been barcoded. So now we are starting to barcode our entire collection and capture the label data in an online catalog. So anybody can go in and search for names or the collector, and that's great. That's providing a whole new use for this type of collection because, as I mentioned, we organize things by species. So if you were looking for a Lewis and Clark specimen, you would need to know the species name. That would be the only way to find it in the traditional collection. But with an online database, you can search that first and maybe get lucky with finding something. We're not quite there. We haven't finished everything. But once we do, it's going to be great. And we're also starting to image everything as well. So now you'll not only get the data, but an image of the specimen to go with the collection.
Do you know how much of your collection you have done this for so far?
A pretty small percentage. All the types, we have about 100,000 types. And we're over on, we're probably close to 2 million now because we have a very high-tech digitization project going on where they're using a conveyor-type system to take images rapidly. So that's really increased the number that we can do in a year. So this is just kind of a fun thing to get everybody excited about plants and dried dead plants. So what do we have here? What's your favorite candy bar? Have you ever thought about all the plants that are actually in a candy bar?
Chocolate. Anything chocolate.
Anything like that. Well, there's a lot of plants in a candy bar, chocolate being one of them. Here's a chocolate pod and a leaf sample pressed on a herbarium sheet. And it's also a good time to point out that we also have illustrations that accompany specimen and plant names. And we actually have an illustrator on staff, and she's very busy because you need an illustration to go along when you describe a new species. So the type specimens that we just looked at, if there's a new name that's being described and you've got a specimen, then there's an illustration that goes along with it in the written description. So it's interesting to have to go along. What else is in a candy bar? Sometimes rice, vanilla. I like the vanilla specimen because this is an orchid, and there are no other orchid species that we really rely on for an edible source. Maybe some leaves, or some flower petals and some kind of fancy salad, but vanilla is a very important economic plant. What else? Here's a peanut, also delicious and important for candy bars. And the other, very important, sugarcane. What would we do without sugarcane? So now you know, next time you eat a candy bar, you'll think about all the plants that are in there.
Okay. All right. So I was wondering how a field person could make use of a herbarium. Why would they come, and what would be the process for them to go through to actually be able to make use of this facility?
Yeah, good question. We have visitors all the time that are coming in to study groups. And the way that we're set up, it's mostly by a family or by a particular species. So, you have space to sort of pull out specimens from the cases, compare things. If you were looking, you may have access. We have a library. You would have access to keys and publications that on particular groups that you're maybe of interest to help you with identifications.
What happens if I find a plant in the field, I press it? Is that something I can bring in? And there's people here that could help to identify that?
So a lot of times, more local, smaller, either institutions or botanic gardens may have people on staff to do some ID. There's usually in states, there's agencies that will help with local ID. Big places like this, it depends. Most of the curators are more specific, focused on groups or families. We do get the occasional, local request or something, and we'll send out pictures around to the department about things. But if you wanted to bring something in, that would be okay. We just have to go through our process of that integrated pest management that I mentioned earlier. So we'd freeze everything. Then you could bring it into the collection. We'd work with you to try to find out, again, using the keys and some of the collection to see what you could match it to.
So would
Kind
you
of like a matching
recommend taking it to say we're in northern Indiana, take it to a local university?
To your local, extension agencies
Extension
or
agency.
what usually So if it's something local, that's probably your best bet. The university's there, any kind of garden, museum that would have a collection like this, or the extension agencies are really good resources for those types of things. But we all have these types of collections. Say if you get an answer there and you want to come to a bigger place like this where you might have more to look at, then that can be arranged as well.
Now you have a lot of multiple samples of the same species, and can you explain why? What would be the reasoning to have that?
Yes, good question. People always wonder, well, if you've got one of something, why do you need multiples? And it's really important because you don't want just one thing at that particular point in time and place in time. Say a species doesn't necessarily grow just in one place, so you want to have a sampling of it from its whole geographic region. And then it's also important to have it maybe over a time span, a time, of that particular locality. Think of that could be used, the data from the label, for understanding flowering times and climate change studies. There's different ways that the data from that specimen could be used to answer bigger, broader biogeographic and biodiversity questions, which a lot of people are really interested in now.
Is there any kind of ethical considerations to keep in mind when you're collecting plants?
Yeah, of course. There's a lot of things. You should never do anything without a permit. Permission of some sort, and that's getting a lot harder. So it is harder for people to go out and just collect anything they want. And there's a lot of things that are listed, that are federally listed, that are state listed maybe, it's an international concern, and all of those things can be found on various websites. And that's another resource. If you were interested in collecting, go to your local institution and find out about the proper steps to take before just going out and collecting. State parks, national forests, you're not allowed to do that unless you have permission from people, and on private land as well. So that's a very good thing to know before getting started.
Can you tell me a little bit about this sample here?
Yeah, so we have other things here as well, not just your traditional vascular plants. that represent, vascular, non-vascular, monocots, dicots, gymnosperms, ferns. We also have an algae collection, and here's a nice pressed example of a red algae. We also have mosses and bryophytes that are kept in small packets like this. Let's see, this one was collected in New Zealand, actually. So again, things from all over the world sampled. And then we have this really beautiful lichen that was collected by a former curator, Mason Hale, in Panama. And it's really retained its color very nicely. It's really beautiful.
Which is unusual, correct? For most plants that are pressed, at least flower color and even foliage color can fade or
That's true.
change color.
And people get so, upset about that. Well, why isn't the color there? Well, there's a few colors. Sometimes purple will last a little while, but yellows don't. Reds will last a little bit. The cactus that we saw earlier. Mm-hmm. But once you start drying out the specimen, the coloring just doesn't stay. So one way of fixing that is to collect flowers. A lot of times orchids or really intricate things might be collected in some sort of alcohol in the field. So it's in a vial of like ethanol that's preserved as a liquid collection. So that can be sort of an ancillary thing to your pressed voucher. We also have, wood samples that will be associated with particular groups. So you're studying pines or something and you want to have big pine cones, you want to have a herbarium sheet with the needles, and then maybe you want to have a cross-section of the wood sample. All of those can complement the specimen.
Okay, well, I want to thank Melinda for a job well done.
Thank you.
There's a lot of good information.
Hopefully.
And hopefully our two upcoming botanists here are off to a good start. But we really appreciate the Smithsonian allowing us and inviting us to come here to share actually a national treasure.
Yeah, thank you for coming. Yeah.
Melinda, this particular seed reminds me of something, and I can't quite put my finger on it.
Yeah, well, a lot of people commonly call it the butternut.